Welcome. Today, we're diving into the critical role of SIP, that's steam in place, in maintaining sterility assurance. We'll explore how this process ensures cleanliness and safety in medical and pharmaceutical environments, and why it's absolutely essential for protecting patient health. Let's get started. Let's start by understanding why sterility is absolutely crucial in pharmaceutical manufacturing. If sterility isn't maintained, the consequences can be severe, from patient harm to costly product recalls. It can even lead to regulatory penalties, production halts, and damage to a company's reputation. That's where sterilization in place, or SIP, comes in. SIP offers a reliable way to sterilize equipment without taking it apart, keeping microbial control consistent. Importantly, regulators expect sterility to be built into the process itself, not just tested at the end. Let's start by understanding what sterility assurance really means. It's about having confidence, the assurance, that a product, process, or equipment system is free from any living microorganisms. The key goal here is to minimize the risk of contamination throughout manufacturing. To achieve this, several components come into play: facility design, how personnel work, environmental monitoring, cleaning and sanitization, sterilization processes, and ensuring container closure integrity. One critical control measure is SIP, steam in place, which keeps process equipment sterile before manufacturing even starts. When it comes to sterilize-in-place systems, there's a clear regulatory framework guiding us. Key authorities include the European Commission, the FDA in the United States, the World Health Organization, and industry groups like the Pharmaceutical Inspection Cooperation Scheme, the Parenteral Drug Association, and the International Council for Harmonization. What they all expect is straightforward. SIP systems must be scientifically designed, validated thoroughly, continuously monitored, and periodically requalified to ensure ongoing safety and effectiveness. Let's talk about sterilization in place, or SIP. It's a validated process that sterilizes equipment right where it's installed, no need to move anything. This usually involves saturated steam or other approved agents to ensure everything is completely sterile. You'll find SIP used in bioreactors, fermenters, holding tanks, and even filling machine pathways. The main goal: to create and maintain a sterile environment before manufacturing starts, keeping everything safe and contamination-free. Let's look at the results of this process. The goal here is to destroy both vegetative microorganisms and those tough, highly resistant bacterial spores. We achieve this through moist heat sterilization, using saturated steam. The key parameters are temperature, pressure, and exposure time, typically between 121 and 134 degrees Celsius, 15 to 30 psi, and 15 to 60 minutes. This combination works by coagulating proteins, denaturing them, disrupting cell membranes, inactivating enzymes, and ultimately destroying spores. That's how sterilization happens at a microbial level. Let's talk about sterility assurance level, or SAL. It's a way to measure how effective sterilization really is, specifically, the chance that a living microorganism survives the process. The industry standard sets this probability at 1 in a million. That's a very high bar. For validated SIP systems, demonstrating an SAL of 10 to the -6 is a must. This level of assurance gives us solid, quantitative proof that sterilization is working as it should. Let's walk through a typical SIP process flow, step by step. It starts with completing the CIP process, cleaning the equipment thoroughly. Then, we prepare the equipment for sterilization, making sure everything's ready. Next comes air removal, followed by introducing steam to begin sterilization. We hold the sterilization phase at the right temperature, carefully monitoring it all the way through. After that, controlled cooling takes place, ensuring sterility is maintained before the equipment is finally released. And remember, every step must be documented and traceable, following strict GMP principles. When we talk about regulatory expectations, it all comes down to keeping critical process parameters, or CPPs, within validated limits throughout the sterilization cycle. These parameters each play a key role in ensuring sterility. Temperature controls microbial kill. Exposure time guarantees the process is long enough. Steam quality affects how well heat transfers. Pressure keeps the steam saturated. Air and condensate removal prevent cold spots and ensure uniform sterilization. Together, these factors form the backbone of sterility assurance. When we talk about SIP validation, we're focused on proving that the process reliably delivers the sterility we need. To do this, we follow a series of qualification steps. First, design qualification checks that the system is designed properly. Then, installation qualification confirms everything is set up correctly. Operational qualification tests that the system functions as expected. Finally, performance qualification shows the sterilization works consistently in real-world conditions. Together, these steps provide the documented proof we need for routine use. Let's talk about biological indicator studies, a key way to verify sterilization. We use biological indicators, or BIs, which contain a specific organism. The most common one is Geobacillus stereothermophilus. Why? Because it's highly heat-resistant and has a predictable destruction profile. For example, we might start with 1 million spores. After the sterilization process, no microbial growth is observed. That means the cycle worked, sterilization was successful. Let's look at the validation results for the SIP cycle in our vaccine manufacturing vessel. Every temperature sensor hit the target. No cold spots were found. Biological indicators came back negative, confirming sterility was maintained even after cooling. With these results, the SIP cycle is approved for routine production. The key parameters were 121 degrees Celsius, held for 30 minutes, at 18 psi pressure, all in a 5,000-liter formulation vessel. This gives us confidence in the processes, reliability, and safety. Our goal here is to build multiple layers of sterility assurance, a safety net that catches risks before they become problems. We're looking at common sterility risks, like microbial contamination, and matching each one with a control measure. For example, validated sterilization cycles tackle microbes, while effective cleaning before sterilization stops biofilms. We also remove air pockets, monitor temperatures to avoid cold spots, design proper drainage to prevent condensate, and use sterile hold procedures to avoid recontamination. It's a comprehensive approach, covering every angle. Looking ahead, SIP technology is evolving fast with some exciting innovations. Smart SIP systems are becoming more intuitive, while real-time steam quality monitoring ensures every cycle hits the mark. Automation is stepping up with cycle verification and digital twins, creating virtual replicas for better control. Artificial intelligence is optimizing processes, predicting maintenance needs before issues arise. Continuous process verification and Pharma 4 integration are setting new standards. Together, these advances point toward a future of continuous sterility assurance, real-time monitoring paired with intelligent control, keeping processes flawless every step of the way. Let's wrap up by looking at why sterilization in place, or SIP, is absolutely critical. SIP ensures manufacturing equipment stays sterile, preventing any microbial contamination. It supports aseptic processing, meets strict regulatory standards, and protects both product quality and patient safety. With documented sterility assurance, SIP enables reliable, consistent, sterile manufacturing. Regulators expect these systems to deliver validated sterilization and maintain sterility throughout use. In short, SIP is the foundation that keeps sterile pharmaceutical production safe, compliant, and trustworthy. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you. Take care, and we look forward to connecting again soon. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.